There are three theories that are important for you to understand regarding muscle contraction: the sliding filament theory, the excitation-contraction coupling theory, and the skeletal muscle innervation theory. Skeletal muscle fibers begin contraction with the release of their internal stores of calcium. That release of calcium is under the control of the nervous system. The neuron communicates with the muscle at a junction called the neuromuscular junction. A neuron stimulates the muscle fiber through a series of steps. These steps are as follows. At the synaptic terminal of the neuron, vesicles store a neurotransmitter called acetylcholine. The neurotransmitter is a chemical messenger. There is a space between the synaptic terminal of the neuron and the motor end plate of the muscle. This space is called the synaptic cleft. An enzyme called acetylcholinesterase is contained in this space. Acetylcholinesterase breaks down acetylcholine. On the muscle, at the motor end plate, there are receptors for acetylcholine. The neuron will communicate with the muscle when an action potential or electrical impulse travels down the axon of the neuron and arrives at the synaptic terminal. When the action potential reaches the neuron's synaptic terminal, changes in the membrane will trigger the exocytosis of ACH into the synaptic cleft. Exocytosis occurs as the vesicles fuse with the neuron's plasma membrane. Acetylcholine molecules diffuse across the synaptic cleft and bind to acetylcholine receptors on the surface of the motor end plate on the muscle. Acetylcholine binding alters the membrane's permeability to sodium ions. Sodium, which is more highly concentrated in the extracellular fluid than inside the muscle cell, will naturally diffuse into the sarcoplasm. The sudden inrush of sodium ions results in the generation of an action potential in the sarcolemma of the muscle. In the synaptic cleft, acetylcholinesterase quickly breaks down the acetylcholine on the motor end plate and in the synaptic cleft, thus inactivating the acetylcholine receptor sites. Once an action potential has been generated in the sarcolemma, the excitation-contraction coupling will occur. As the action potential travels through the T-tubules, it will trigger the release of calcium from the cisternae of the sarcoplasmic reticulum. The calcium surround the sarcomeres and almost instantly binds to the troponin on the troponin-tropomyosin complex. Calcium ions bind to troponin, weakening the bond between actin and the troponin-tropomyosin complex. The troponin molecule then changes position, rolling the tropomyosin molecule away from the active sites on actin. When the active sites are exposed, the myosin head can now interact with it. Once the active sites are exposed, the energized myosin heads bind to them, forming cross bridges. The myosin head has already been activated, and when the myosin binds to the actin molecule, the myosin head pivots toward the M line. This action is called the power stroke. When this occurs, the ADP and the inorganic phosphate that was bound to the myosin head is now released. After the power stroke, another ATP is now able to bind to the myosin head. When this ATP binds to the myosin head, the link between the myosin head and the active site on the actin molecule is broken. The active site is now exposed and able to form another crossbridge. Myosin can be reactivated when the free myosin head splits ATP into ADP and inorganic phosphate. The energy released from this hydrolysis is used to recock the myosin head. This is an example of a resting sarcomere where each myosin head is already energized or charged. See how each myosin head is pointing away from the M line. In this position, the myosin head is cocked. This diagram shows the contracted sarcomere. The entire cycle is repeated several times each second as long as calcium concentrations remain elevated. Calcium levels will remain elevated as long as action potentials continue to pass along the T-tubules and stimulate the terminal cisternae to release calcium. A contraction will last as long as the action potentials continue in the neuron, as long as the free calcium ions are present in the sarcoplasm, and as long as ATP is available.